Hello everyone, this is Women in Science Day. I'm Professor Carmen Inda from the Natural Science Department at Hostos Community College. I would like first of all, because this is a session where we're gonna have a lot of people, that you mute your microphones, please, so we can all listen to each other. So as I said, for those of you that just joined, I am Professor Inda, Professor Carmen Inda from the Natural Science Department. I have been organizing this series of events of Women in Science from a couple of years already, and it was regarding challenges of the women that we have been facing during our careers, and we would like to help other students, and we would like to support everyone in the different stage of their career. In this specific situation, we decided that it was very important not just to talk about career challenges, but also about the crisis situation that we are living these days. So I am a scientist, and my colleague scientists, they are gonna be giving a beautiful presentation, and they will address the students' concerns about coronavirus, how to be protective, what are the measurements that we are going to take, how are we going to be safe, what are the treatments from this for this COVID. So I'm going to start sharing the screen so you can see the presentation that we are doing, and I would like to thank everyone to be here today. And please, Maria, receive my apologies if this process doesn't go smooth as smooth as we would like to. But this is the first time I do a session online, and I hope that we can have the best outcome. Let me just. Exactly. So here is the session that we are holding today. This is the Women in Science activity. If you would like to join, you have some other friends that would like to join, you can Zoom, you can do it using Zoom, okay? First of all, I would like to say, so I just introduced myself. My name is Professor Carmen Inda from the Natural Science Department at Hostos Community College. I would like to say that, first of all, this activity is in memory of our dear Jaime Luján, who lost the fight against COVID-19. We are missing him a lot, and all his contribution to the department was amazing. All of the students miss Jaime, and all of the faculty really miss him. So I'm gonna share a video that Karin Contreras did in memory of Jaime. We have here also some family members from Jaime, and I would like to say that they join us in this activity today. And I would like to say in the name of all the department from Natural Science that we would like to send our love, our condolence, and as Eva always say, Jaime will always be with us. I want to thank Karin Contreras that she put this video together. We're gonna start. So I'm going to start this event in memory of Jaime Lujanson. We are gonna have this event in 3 sessions, okay? We're gonna have 3 sessions in this Women in Science Day. The first is gonna be listen to real expert advice on coronavirus. We will have Dr. Maite Sánchez Aparicio from Mount Sinai School of Medicine, and we will have Elizabeth Diego Navarro from the New York City Department of Health. In the second part of the session, we will have navigating career challenges. As professional women in science, what would we do? What women in science during the COVID-19, okay? Now, what? How to adapt the changes. All these professionals will speak about their careers, the skills and that they got, and how they are responding to changes. Dr. Sara Martucci from Mercy College will be in this panel. Dr. Svelana Karilov from the Myeloma Research Foundation. Arlie Whisper, who is a former student from Hostos Community College, is going to talk about her career, and she's an associate engineer at Merck. And Yaya Rajwani, she's an assistant professor at Hunter College. We are going to have in each of the panel, in each of the sessions, we're going to have presentations and questions from the students. First of all, we're going to ask questions that the students prepared previously, and after that, we are going to have, we will open for a couple of minutes the microphone to each of you that we would like to speak. Then at the end of the session, because during this difficult time, we need advice on several issues, we have 2 professors from Hostos, who are Inmaculada Lara Bonilla from Humanities Department, and Professor Lisette Colon. She's a counselor and she has great experience on our college on counseling. So they will both give a presentation with specific tips and advice that they're giving in these specific times. We will have the greeting remarks from our provost at 2 p.m. And I would like to thank the Office of Academic Affairs and Women's and Gender Studies Committee for their support. As I said, this activity is in memory of Jaime Lujan. And also, we have to, and also we are going to have a final presentation at the end, right? Okay, so I would like to introduce briefly our students that they will introduce our speakers, okay? So this event, during this event, I'm gonna be the organizer and moderator, but my students from Biology 210 and they will, Bio 230, they have been participating in this event, preparing questions, and I'm gonna be introducing our speakers. Hello, Sadia, hello. Alex, and hello, Maria Fer. So, Maria Fernanda Armijos, hello. Thank you very much for being here. And thanks to all of you guys. Thanks to all of the... Okay, so, Maria Fernanda Armijos, Faqir Al-Sarahat, sorry if I don't pronounce it properly, and Sadia Zaman, they are 3 of my students from biology, and they are going to be introducing the speakers. I'm going to pass the word, first of all, to, let me see, to the presenter of Elizabeth Diago, who I think is Sadia Zaman. Hello, Sadia. You can go ahead. Sadia?
Oh, hello everyone. So I was going to talk about Dr. Diego Navarro today. So she holds a biomedical sciences PhD degree and a public health master's degree Currently, she works at the NYC Department of Health as Ebola and Special Pathogens Program Manager. Her academic research focuses on developing novel strategies to treat and diagnose antibiotic-resistant infections. She currently manages New York City's healthcare system preparedness effort for managing high-consequence infectious diseases. And she is interested in One Health approach and recognizes how humans and animals' health are linked to environmental health. So Elizabeth is also co-founder of ECUSA and co-chair of MECUSA, which is committed to pursue gender equality in science.
Thank you very much. Thank you very much, Sadia. Thank you, Elizabeth, for being here with us today, Dr. Diego Navarro. Thank you very much for being here. We are waiting for your, we're looking forward to your presentation.
Thank you for giving me the opportunity to be here. So now let me try to share my screen. I know you have to stop your...
I already pinned your video. Yeah. You're there.
Okay, now. So, oops, wait a minute. I just went to the, okay. So yes, first I want just to thank you to inviting me to give some, to talk about COVID-19. And I just want to share that the things that I am going to talk is not in the name of the New York City Department of Health, but I will give you my best advice on the questions that you will have and what I am going to present.
Thank you.
Yes. Yeah. So what I am going to talk to you now is very briefly, so you would understand why we are doing all this New York State on pause. So you would understand a little bit of epidemics concepts and the measures that we are taking in order to control the epidemics, why we are doing it. So one important thing that, why is not? Okay. So one of the most important things in the epidemics is the reproductive rate of the infectious disease. So here, this means how many people would be contracting the disease for each individual that is infective. And this is going to depend on the probability of transmission in a contact between infected individuals and susceptible ones. So if you are very close to someone, you will have more probabilities of transmitting the virus to another person that is susceptible to be infected. The frequency of contacts in the populations, this will depend on the social patterns. For example, if you are in a, if you are contacting, for example, in a crowded subway, you will have more frequency of contacts in the population that if you are isolated at your home. The duration of the infectiousness, so this would mean the period from where you are infected and you start shedding the virus, so spreading the virus until you are completely cured. The longer that is this period, the longer probabilities you will have to transmit that virus to another person that is susceptible. And also it would depend on the proportion of susceptible versus immune in the population. I will explain this concept later on. So the R naught, or is the basic reproductive rate. And this is the number of people that one infected person transmits the virus in the absence of immunity. This would occur in this case with a new virus when it comes into a population and it is the beginning of an epidemic. When this virus started to spread in population in China or in any other parts of the world, all of us, we were susceptible to be infected because there is no known immunity to this virus because it's a new virus. And later we have the effective reproductive number. And this is the real number of persons at any point during the epidemic that an infected person transmits the virus. This is going to be changing over time, because at the beginning, for example, all of us, we were infected, we were susceptible, and there were not measures to control the epidemic. So this number was much higher than now. If we are at home and we don't have any contact with other people, the number would be decreasing. So this is very important, because when this number is less than 1, it means that the disease is going to disappear. If you understand, less than 1, that would mean that one person that is infected is going to infect less than one person. So in a pool, there would be numbers of people that would be getting infected, but one person infecting less than 1, it will mean that in the end, the disease will disappear. If a person infects another person, that is the R equal to 1, the disease will continue in the population. And when the R number is more than 1, so when one person infects more than one person, this means that the disease would become an epidemic. And this is what we have been seeing when the peak of contagious cases it was increasing over time, because we were growing. This R number was growing. So this effective reproductive number, yeah, it depends, as I said, in the proportion of susceptible people and immune people in a population. What it means. At the beginning of the epidemic, people in the population were susceptible. They were non-immunized. There was no memory or there was this virus was new, so everybody was susceptible to get infected. So if one is infected, all the people in the population would be could be getting sick. But as the epidemic progresses, because this is a dynamic situation, some people that got infected would be recovered and they would be immunized. And the proportion of people that can get infected would be decreasing. And also, sadly, there are people that in the population that get infected and doesn't recover because they die. So this also decreases the number of susceptible people to get infected. And how this immunity can be achieved, it would be through vaccination, because a vaccine, what it's going to do is to protect you to get infected, but also through recovery of those sick individuals. So in the end, when we achieve a threshold that is like the herd, it's called herd immunity, it means that even if there are some susceptible people still in the population, as the virus cannot be transmitted easily between the contacts, these susceptible people would not get infected. So, as I said, this would be achieved through vaccination and recovery. And the vaccines that we usually receive when we are children, what they want to achieve is to get this herd immunity, so those virus or bacterial diseases don't get spread in the community. But also, this effective reproduction number depends on measures that reduce the possibility of transmission affecting social and environmental patterns. These measures could be targeting the disease control by testing and identifying very quickly those individuals that are infected, and they would be isolated. So you would be reducing the time that those people would be making contacts with susceptible people. Also, by contact tracing, this would mean that once that you find a person that was infected, you trace all the contacts or close contacts that that person had, and you isolate them in order, or quarantine them to see if they develop the disease. And also infection and prevention and control in healthcare settings. So all the washing, all the the protective equipments that healthcare workers were using when they were caring for an infected individual in the hospitals. And later there are non-pharmaceutical interventions. So these are the ones that we are able to, or the community is doing right now. So for example, the physical distancing. This is when the New York Post guidelines are saying that you should stay at home as much as possible as you can. Later, there are individual interventions, something that we as an individuals, we can do in order to protect ourselves and to also reduce the virus that we may be releasing or shedding. So this would mean washing our hands frequently, cover cups. So as you have seen, I'm sure that you have seen, we shouldn't be coughing in our hands. We should be coughing in our elbow or in a tissue that later we throw away. And the recent guideline to wear masks. These masks are not meant to protect you, but are meant to protect people in the community if you are sick or if you are infected and you are shedding the virus. Later, there are environmental interventions. So these are clean surfaces and increased ventilations in offices or in classrooms, at home. So for example, cleaning the subway. These are things that you can do in the environment to control the virus that could be available for other people to get infected. And later there are lastly, the community interventions. These are interventions that you do in the community as a whole. For example, social distancing of high-risk groups. So in this case, elder people, or closing schools, or the general social distancing guidelines that we are hearing. So you should be, if you go outside, you should avoid to be close to another person in a space less than 6 feet. So all of this is really nicely put in a website that is, this is an NGO that is called Prevent Epidemics, and they are meant, this organization works to prepare different states in order, different countries and states to be better prepared to respond to epidemics. And they did this nicely figure where you would be seeing cases over time. This is the line. So right now, when all these New York State on post guidelines were set up, it was to flatten this curve. So they wanted that this epidemic peak to be as low as possible. So the cases that come into the community in whole, because all of us, we are susceptible, to be as flat as possible. This is the flatten the curve. And at the beginning of an epidemic, you can do containment strategies, that is to contain the virus in order that the virus doesn't doesn't spread in the community. You do this at the beginning by testing people and tracing the contacts. But later, when we were in flatten the curve state, we are doing the mitigation. We will try to mitigate the bad effects that the epidemic is posing in the community, and more importantly, in the healthcare system. And soon we are now seeing like there is a decrease of new cases, so we will be entering in the suppression phase. as you see, all these non-pharmaceutical interventions, the personal, the environmental, and the community should be going. But this would be a stage where it could be going into a tighten strategy or a loosen strategy, depending if the cases go up again or go down. And this is going to happen until we have an effective vaccine, in order that we make people we make people immune to the disease, and the virus would be stopped to be spread in the community. But this is going to take time. So yeah, this is my presentation of, I hope that you understand, but I am open to answer any of the questions that you could have.
Thank you very much. It was really informative. Thank you very much for being here with us today, because it's really, really important to have all this information for all of our students and our faculty. And we're gonna have the following presenter, and then at the end, we will have a session of questions from our students. So let me share my screen now. And we're going to have our next speaker, and we are going to have the introduction from María Fernanda. María Fernanda Armijos is one of our students from Hostos, from Valle Tutén, and she is going to introduce our next speaker, Maite Sánchez. Thank you very much, María Fernanda.
Hi, good morning, everyone. Dr. Maite Sánchez, Aparicio, PhD. She is from Mount Sinai School of Medicine. Dr. Sánchez was born and raised in Madrid, Spain. She studied pharmaceutical and sciences at the Complutense University of Madrid. These studies sparked a keen interest in science, so she decided to join a laboratory research and obtain a PhD in biomedical sciences. Studying one of the most devastating diseases in animals, foot and mouth disease virus, she specialized in virology. She moved to New York City for a postdoctoral position in the Department of Microbiology and Mount Sinai School of Medicine. Here, she focused her research in human infectious diseases such as influenza. She specialized in how dangerous pathogens invade the human immune system and how the human system reacts against it. Along this period, she has published 20 scientific publications. Nowadays, she continues her studies as a research assistant professor at Mount Sinai School of Medicine, understanding how new pathogens such as Zika, HIV-1, and COVID-19 can infect humans.
Thank you very much for this beautiful presentation. I'm going to give the microphone to our speaker, Maite. Maite, hi, how are you? I'm gonna. Maite, give me 1 second. Give me 1 second. I think that your microphone was not working. Hello, Maite. Thank you very much for being here with us today. Thank you for your participation in the event and for your beautiful presentation. I'm pretty sure it's going to be very informative. Let me stop the screen, and then I'll let you do your presentation.
Okay.
We hear
Can you hear me now?
Yes, now perfect.
All right. Okay. So first of all, thank you, Professor Carmen, for giving me this opportunity to talk today here. And thank you all for listening. I hope you learned something new. I'm going to talk about more on the virus side of this pandemic. And this is the outline of the presentation. I'm going to talk about what is a coronavirus, what are the symptoms, how to prevent the infection transmission very fast, because Ellie did a great job explaining it. And then what we are currently doing in research and all the pharmaceutical companies about new antivirals and vaccine strategies. So first of all, what is a coronavirus? A coronavirus is a virus that belongs to the family Coronaviridae. It has a positive single-stranded RNA of about 30 kilodaltons. It's of big size, it's 120 nanometers diameter, and it's called coronavirus because corona means in Spanish crown, and this is due because it has the spike, the protein that is in the outside of the virus, reminds us to a corona. That's why it's called coronavirus, and in English it's called crown. We know that so far there is 7 coronaviruses known to infect people. These 7 coronaviruses can be split into groups. The human coronaviruses that cause the common cold that we all have had at some point. And these are alpha or beta coronaviruses that we can hear with these weird names, these 4. And then we have the other group of coronaviruses that infect people that are zoonotic coronaviruses. These zoonotic coronaviruses spread from non-human animals, usually vertebrates, to humans. So the problem of these new coronaviruses is that we don't know the degree of lethality or how it's gonna affect our immune system, because we never saw these viruses before, because they jump directly from animals to humans. In this group, we have SARS coronavirus 1, that it was originated in bats and it jumped to humans in 2010. And then we also have MERS in 2013, that the origin was in camels. And in this group, we have the new coronavirus. And I'm gonna show you a little, very briefly the replication cycle of a coronavirus. So we have it here. This is the virus, and this is the cytoplasm of a cell, and this is the nucleus, we cannot see it, sorry. So the coronavirus with the spike protein, that is this one in green, can recognize the receptor which has to bind in order to enter into the cell. This receptor is in the membrane of the host cell of our cells. Then it's recognized, and then the virus releases the viral genome. This viral genome is going to be translated, is in the RNA, it's going to be replicated, and it will make more proteins, which will be assembled and will make the new virus that will be released from the cell to infect new cells, new hosts. So that is how, in general, works the replication cycle of this virus. So this new virus.
Maite, one question. So the RNA takes taking control over the ribosomes, or it goes to the nucleus of the cell?
The RNA is usually in the cytoplasm of the cell only, but takes over all the replication machinery of the host cell. So somehow it's sequestered in order to replicate itself and to make more virions.
Mm-hmm. Thank you.
Okay, so this novel coronavirus, I guess you all know, the first case was in Wuhan, in an area in China, and you can see here the map. And it was reported in December 31st of 2019. And we think that the cases were related with the wholesale market in Wuhan, where many people is in very close contact with animals. Since December 31st, this coronavirus has been spread to more than 187 country regions. As you can see here, in this map, you can show with the red dots where we have cases of coronaviruses. And we have a total worldwide of more than 3 million with the total deaths of more than 200,000. And this website is very good. If you are interested in numbers or in data, you can always check it out every day, because it keeps changing. So what is the name that we are having for this new virus? So we have 2 names. The virus, that is called SARS coronavirus 2, because it's very similar to the first SARS one. And the disease is gonna be called coronavirus disease 19, because it occurred in 2019. It started in 2019. So the symptoms that we have with this virus can be very non-specific, and many of them are asymptomatic or can develop flu-like symptoms such as fever, cough, difficult breathing, or if you have a more severe case, you can have respiratory diseases, coughing up blood, or even kidney failure or more things. No? The problem of this disease and why it started to spread so fast is because one of the things is because the symptoms can appear as many as 14 days after you get infected. So you have 14 days that you are asymptomatic and you are in contact with people, and then you can infect many people without knowing it yourself. So I'm not gonna go through this because Ellie explained it very well, but that's one of the problematics, that we have many cases in one certain time. So what we can do is to flatten the curve, trying to help the hospitals and the doctors to not be overwhelmed and to have the capacity to treat everybody that has the virus and can go to the hospital. So this is a slide on how to prevent the COVID-19. Eli also talked about it. So what we can do as a community is to wash our hands, maintain social distancing, avoid touching eyes, nose, and mouth, practice respiratory hygiene, and if you have fever, cough, or difficulty breathing, you have to go to the doctor in order to be tested and cured. So this is another slide that summarizes everything. I just want to point out that the virus is transmitted by respiratory droplets. That means that the virus is alive in your saliva, for example. So when you are coughing or you talk with someone, we always have this kind of microparticles that can go out of our nose or our mouth and can go to a new person. That's why these droplets survive only, can go as far as 5 feet or something like that. So that's why the distant measurements are 6 feet apart. And how our immune system responds to this virus. We have different stages once we start to be infected. We have the window period that is called, is where we are asymptomatic. And then about day 7 to 14, or depending on the person, the symptoms start to appear. And then the patient begins to recover. And then you have what we call convalescent patients that are already recovered. So what happened during this time? We can detect a virus, RNA of the virus, since the beginning of the symptoms until approximately when the patient starts to recover. In the meantime, about 1 week after we are infected, we have an antibody response that is called IgM. What it does, it starts to appear, and then also it fast declines, as you can see here, around day 20. This is an average. Sorry. And then we have another kind of antibody response that is called IgG antibody response that starts to appear right when the patient starts to recover, and is the one that can provide that more long-term immunity. So how can we detect all these kind of curves with the test that we have? We have 2 different tests, the viral test and the antibody test. The viral test only tells us if we have a current infection. As you can see here, this will be the blue line, because it is only detectable until day 20 or something like that. And then we have the antibody test, and as I told you, is only the antibodies are only detectable when you have had a previous infection. And this can take even up to 1 to 3 weeks to make antibodies, and your body to make antibodies after the symptoms occur, as you can see here. So between these two, the green IgM and the red IgG, the best is to have IgG's antibodies, because this means that you will be protected against the virus. It's like if you were to have a vaccine. So this is a slide to explain you a little bit how the viral test, the test that detects viral RNA, works. So what usually we do is we take a cotton swab and we put it in the nose, and then this cotton swab is gonna be stored or mixed with a buffer or a dilution, and then from there we will extract the viral RNA. This viral RNA will be modified to obtain cDNA, and this will be amplified by qPCR. This technique needs very specific primers and probes from the virus that only recognizes this specific virus and no another coronavirus, for example. And then you will have the test results that can be positive, in red, or negative. The other kind of tests are the antibody tests, and these are called ELISA. And very briefly, how you can see here, what we need in this case is blood sample. So we take some blood of the patient, we load it in with a buffer, and then we run it in a kit or in a slide where we have antigen of the coronavirus conjugated with a fluorophore that can we detect that we can visualize by eye. So if we have antibodies against the virus, and these antibodies recognize the antigen of the virus, we will form a complex, and you will see the red line here. Okay, and then you have all the controls that you need. So the other way to try to stop the pandemic or the number of the cases are the treatments. As Ellie said, these are more long-term, and it's gonna take a while to get them, but we are getting there. There is many groups in the US and around the world that are working in getting new antivirals and new vaccines. So there is two main strategies right now that all the research groups and pharmaceuticals are working on, and this is based on the viral entry mechanism of the virus. So as I told you before, this virus uses a receptor in the cell called ACE2 to enter into the cell, and this receptor is recognized by the spike protein that is here in purple or in blue. So the strategies are to target either the ACE2 receptor in our host, in our cells, by doing antigens or antibodies that bind to it, so the virus cannot bind, and then the infection is prevented. The other strategy is to target the spike protein of the virus or the receptor, of the receptor binding domain of the spike of the protein, and we have the same result. The virus will not bind to the receptor of our cells, and then we will prevent the virus, the infection. The other treatment that has been approved, I think it's in the news now by Dr. Fauci, I think he said, they released it, this is called remdesivir. It's a drug that was repurposed to treat COVID-19. This is based in another mechanism different to the ones that I showed you before. This is a nucleoside analog, and what it does, it interferes with the replication machinery of the virus by blocking the polymerase, that is the one that makes new RNA of the virus. And therefore, it inhibits the RNA replication of the virus and stopping the replication. And that's how it works. This drug was developed like years ago to treat Ebola, and it didn't work for Ebola, but then what they saw is that it can work for this new coronavirus. And I think we're gonna have very good results. The other kind of treatments that we have right now, and the best that will give us long-term protection, is vaccines. The vaccines are gonna take, and usually they take much longer than antiviral drugs, because you need to have a candidate, and then you have to take that candidate and go to clinical trials testing. And this is gonna take at least 12 to 18 months. So I show you here a schematic view of the approaches to viral vaccine development that are going on right now. Usually, in the most common strategy is to use the spike protein, the one that is forming this crown in the virus, and use it as the immunodominant epitope to develop antibodies against that. We have one strategy is, for example, live attenuated viruses, where we immunize people with the viruses that cannot replicate, for example. Whole inactivated viruses are dead viruses, or split inactivated, that is only one, the virus is split, or synthetic peptides, virus-like particles. One of the strategies is DNA or RNA that is now giving a good proportion of immunized people. So let's see what happens. It's now in clinical trials, I think. We have the other strategy that is recombinant subunits, recombinant bacterial vectors, or recombinant viral vectors. So this is an example of 5 clinical phase vaccine candidates for COVID-19. As I mentioned before, this is called Moderna. This is made of mRNA with the spike protein encapsulated inside. This is also using the spike protein in a DNA plasmid expression vector. This is using also the spike protein in an adenovirus type 5 that expresses this protein. This is using dendritic cell modified for antigen-specific cytotoxicity lymphocytes. And this is also using inactivated virus vaccine, the whole virus produced from viral propagation in cells infected with SARS coronavirus clinical strain. So far, this is a summary of what is going on in the research field. And hopefully one of these will help us to fight against these viruses and to get a protective immunity for all of us. So I'm gonna stop here, and I want to thank you all for listening, and I will take any questions you have. Thank you so much.
Thank you very much, Dr. Sánchez Aparicio. Thank you very much.